Thank you for the kind invitation and the kind introduction, and thank you all for joining my talk. As Chris said, I'm Carlos, and I'm a last year PhD student at the Oxford Protein Informatics Group. And I'm going to be telling you about some research that I've been carrying out for the past months, trying to understand if the methods that we have for structure prediction can actually be used to understand something else, which is how proteins actually reach this native state that they need to perform their function. I think, well, I don't need to talk about this because John Mould already gave a fantastic talk about it, but this all started in CASP14 in December when AlphaFold achieved these wonderful results where they can essentially predict almost anything with unbelievable accuracy. It was personally incredibly, let's say, impactful. It made a huge impact on me. I remember after the first day of CASP, I was quite literally devastated. I was wondering, oh my God, what am I going to do now? All that I was hoping to do for my PhD is actually now not interesting anymore. So I spent an important amount of time doing soul searching and trying to think what questions are going to be important from now on. Now we are as close as ever to a solution to the protein structure prediction problem. And I spent the best part of that week writing a huge blog post where I talked about what had been achieved, what I thought was important about it, and where I was seeing that there will be impact from AlphaFold in bioinformatics, in molecular biology, and in pharmaceutical discovery. And based on this reflection, I realized that the problem that I wanted to look at then was protein folding, which is what I'm going to be talking about. There are a lot of reasons for this, and one of the speakers already introduced what it is, why it's important, and what the challenges are. But in particular, the reason why I think it's really crucial to look into this is because even though protein structure prediction is going to help us design new pharmaceuticals, because it's going to give us structures that we can dock things into, we still don't know a lot about many diseases in terms of the targets that we need to drug. So things like Alzheimer's, for example, that even the recent approval of a pharmaceutical by the FDA, we still don't quite understand how it works, and we don't seem to have any really effective treatments for it. So that is based on ultimately on protein folding. And by understanding protein folding better, we might be able to find solutions to these diseases and many others. It's also an incredibly interesting scientific problem that's been open for nearly 60 years, I think. So I'm going to tell you what I actually did. So I designed an experiment in which I took 6 state-of-the-art protein structure prediction programs. If you're interested, those will be Rosetta, which is the classic fragment replacement program. Same2, which is a code that we wrote in my lab that also does sort of fragment replacement with some other tweaks. I looked at EVFold, that's authored by the Marx group at Harvard, that uses well, essentially contact prediction and CNS. I looked at DMP-Fold that uses deep learning, and I looked at RaptorX and TRosetta, that both of them use deep learning to predict the distances between interresidue amino acids, but also relative torsion angles. I spent some time looking into these codes, and I modified them so they will print the trajectory of folding as they predict the protein. And I spent some good time looking into the literature to collect a database of protein sequences for which you have experimental data. I collected about 170 proteins with different levels of annotation. So for some of them, we only have kinetics, what is the sort of folding pathway, but for others, we have hydrogen-deuterium exchange experiments that tell us, well, there's an intermediate in this folding pathway, and actually, this intermediate looks like this. So there are very variable levels of granularity in what we have. I used the modified state-of-the-art protein structure prediction codes, and I predicted the structure of all of these protein sequences for which I have data. Well, some of the programs are a lot more expensive to run than others, so I used variable numbers of decoys, but roughly I used about 200 decoys for the simplest programs and about 10 decoys for the more complex ones. And I spent a very long amount of time trying to understand how to make sense of tens of thousands of protein structures. After trying a bunch of stuff, I realized that, well, actually, all of these trajectories were quite unphysical. They had a lot of problems. Sometimes they don't look like anything that you will expect to happen really in solution. So what I did was take a very pragmatic and simplistic approach. I reduced the data to the fraction of native contacts between secondary structural elements. So I took my proteins, I ran them through STRIDE or DSSP. identified what were the main secondary structural elements, mostly alpha helices and beta strands, and then computed the structure of native contacts between this. And then finally, after this pipeline, I used a very simple time series analysis to identify which phases of folding were present in these trajectories. And from those, you can also identify, well, this phase of folding is, for example, when this alpha helix gets in touch with this beta hairpin. You can get a very sort of coarse-grained, but also simplified view of what is happening in the trajectory. So then I took all of my analysis and I started to compare it against experimental data that I had gathered before. So for example, the first experiment that I ran was to try to predict the folding kinetics of the different proteins. Historically, the folding kinetics have been grossly separated into two main classes. We have the two-state folders, which are proteins that just fold cooperatively in one step and are called two-state folders because you have the unfolded state and the folded state. This is something that's mostly present in small proteins. And well, there can be some discussion as to whether it is an actual description of the physics that are happening in there, or it's simply what we can see with our macroscopic experiments. I looked at the phases that I had identified with my methods and tried to see if I could predict these mechanisms using the programs. And what I obtained was the table that you have here. There's quite a lot of information here, and it can be slightly overwhelming to interpret. So let me try to boil it down to the really important information. First of all, you can see that I have the 6 programs that I looked at, and I also have as a reference the length of the protein chain and the contact order. And you can see that I have them only in the rows that say supervised accuracy, supervised F1 score, because these are simply a logistic regression trained on the length or the average contact order of the structure. If you compare all of these metrics, doing, for example, a bootstrap hypothesis testing, you will find that all of these methods are significantly better than a random baseline at the 99% level of confidence. They are also better than the contact order, which is a simple sequence agnostic structural baseline. So it's something that tells us about how the structural modulus looks like, how sort of convoluted it is, but it doesn't tell us anything about which amino acids are interacting with which. And they're also better than that. So they are not entirely random. They know something about folding. They are slightly better than if I actually had the structure, but I didn't know what the sort of amino acids were in there. But the real problem here, and the thing that really made me think, is if you apply the same statistical testing to a linear regression based on the length of the protein, you will find that none of them are significantly better. And in fact, a good fraction of them are significantly worse. So just by taking the FASTA sequence of the protein, counting the number of amino acids, you can be better at predicting the kinetics than an incredibly complex method using multiple sequence alignment, a lot of evolutionary information, a lot of potentials, a lot of parameters trained from a lot of things. And to boil it down, almost everything that we know about protein structure prediction, they are still not better than simply counting the number of amino acids. And well, I think this is just an incredible result. I still struggle to believe that this is true, but I've tested it again and again and again, and it seems that this is how it is. Surprised as I was by this, I decided to go to the second test, which was inspired by a classic paper of Plaxco, Simons, and Baker that another speaker has mentioned before. Essentially, these guys took a set of 12 proteins, and they observed that the folding rate constant was very highly correlated with the average contact order of the protein. I tried to examine whether these methods, all of these protein structure prediction methods, can provide a similar correlation. So in these plots over here, I'm just providing a sort of correlation between the average possession of the folding event in a trajectory. By the way, these are two-state folders, so there's only one folding event. And I'm seeing if it's correlated with the natural logarithm of the folding rate constant inspired to what Plaxco, Simons, and Baker did. First of all, before we go into the models, you can see that the results still hold, even though I examined about 80 to 90 two-state folding proteins, while Plaxco, Simons, and Baker only looked at 12. But in fact, the length of the protein is slightly better than the contact order of the protein, at least in the dataset that I'm looking at. Now, if you go and look into the protein structure prediction programs, what you're going to see is that there is a massive divide between some of the sort of worse methods that's have sperm correlation coefficients that are between 0 and 0.10 and that are in the wrong direction, because this is telling you if your folding event happens later, your folding rate constant is actually going to be higher instead of smaller. The one difference is, in this case, Raptor X, which presents a very nice correlation. You can see this, you can almost get a line over here. It has a sperm correlation coefficient that is comparable and significantly equal to the length of the protein. So we go again to the same thing that we discussed before. We looked at all of these methods, very sophisticated methods. We compared them against a very simplified predictor, and we observed that they are actually not better, and in many cases, they are much worse. So there's, again, something that we have wrong about the mechanistics that we're looking at for protein structure prediction. Well, this was for two-state, so then I decided, okay, what happens when we actually have an intermediate? So I ran the next experiment. This is just a reproduction of the figure that I showed before. And in this experiment, you're looking at different phases of folding that you have. I'm looking at the first phase, which we can identify with an intermediate, and I'm trying to see which interactions are being formed in this intermediate. Then I am taking all the hydrogen-deuterium exchange experiments that I have, and I'm trying to identify, do I get the same intermediates in my trajectories that I will get in an experiment? Are these predicted intermediates consistent with the experiments in folding? And the answer is, I am afraid, and this is becoming a trend, no, they aren't. You can see in this table the comparison of several methods with a sort of random baseline, because we can't predict intermediates using length of contact order, unfortunately. And what you can see is, again, none of these methods is significantly better than what you get with an unbiased random coin. And well, in this case, none of them seem to be worse, but at least they are not significantly better. So this brings me again to the same argument that I've been repeating again and again. We have very sophisticated methods, we have very simplified and well-defined problems, and still we don't seem to be able to reach a solution. In this case, however, I do have an explanation of why they are not that good. So I took all of these predictions and I transformed them into bit strings. So essentially, if you take, say, pairs of secondary structural elements, I assign 0 to those bits when they are not in contact, and I assign 1 to those bits when they are in contact. And I compute the Jakat distance, which many cheminformaticians in the audience might know as the Tanimoto distance. And when you look at sort of pairwise interactions between these secondary structural elements, what you observe is there's an enormous randomness in them. So as a quick reminder, the Jakat similarity essentially means if you have one Jakat similarity, it means that all of your sequences are the same. They are identical. If you have close to 0, because it's rare that you can get to 0, it means you have very, very diverse sequences. In these plots over here, I collected all of the trajectories that had multiple folding phases for each protein. I collected them, and I computed the average Jakat similarity between all of these trajectories. And I represent all of this in a histogram in these plots. And what you can see is there are very few proteins, about 3, 4 perhaps per program, where you get a similarity that is close to 1. To make matters worse, in most of these cases, you actually have very small proteins. So you have, say, 4 pairs of secondary structural elements, then it is very difficult to get a low Jakat similarity simply because there are so few. But in practice, the vast majority of them have almost no consistency whatsoever. And in some examples, you can find that each of, say, 50 trajectories, all of them have different intermediates, which means these programs are about as good as an unbiased coin because they are behaving like an unbiased coin. They just outputting random intermediates again and again. So at this point on my talk, I have told you that protein structure prediction methods are not good at studying kinetics. They are not good at predicting rate constants. They are not good at predicting intermediates. But I have told you this in sort of a very wide, tens of thousands of data sets at the same time. So I'm going to try to zoom in into a very simplified protein folding case that some of you might be familiar with. And this is a ubiquitin folding. So ubiquitin is a very small, I think it's about 80 amino acid long protein, really, really small. It's been studied experimentally very widely since the 90s, and it has the particularity that it was studied computationally by the research group of D.E. Shaw using the Anton supercomputer to run very long molecular dynamics simulations. The folding kinetics of this protein were sort of a heated debate at the beginning of the century, but there seems to be an overall agreement in the experimental community that the protein folds via two-state mechanism. So it folds cooperatively without showing any intermediates. So I took this protein and I compared the folding produced by all of the methods with what we know experimentally, and also with a coarse-grained molecular dynamics force field that I wanted to use as a sort of baseline of what people use computationally in studies of protein folding. This force field in particular was parameterized by one of my colleagues at the University of Oxford, who works exclusively on modeling folding pathways using molecular dynamics. I would like to first draw your attention to this graph over here, this bar plot with the colors. I would like first to take into account on the y-axis, we have the portion of two-state trajectories. So for each of these methods, I generated 200 trajectories, and I counted how many of them are consistent with two-state behavior and how many of them present multiple folding phases. You can see that 3 out of the 4 methods that I'm looking at present a majority of two-state folding. This is actually not entirely surprising. So if you look closely at the DMP fold data, for example, most of the DMP fold trajectories are actually two-state. So roughly 90% of the trajectories in every protein in the dataset are two-state in the DMP fold dataset. Still, in this case, it's in the upper end. So out of 200 trajectories, there's only one that presents multiple folding phases. Tia Rosetta and EBFold are about the usual. So if you compute the set score, for example, you'll find that it's slightly above the mean. So they are actually predicting that it's two-state. And then RaptorX seems to be the outlier here that predicts mostly multiple phases. But obviously, the method that I am using is actually quite simple. So I'm not using anything particularly sophisticated. I'm just looking at big changes in the protein. So it might well be that there are some features of the unfolded states, for example, that I'm looking at that I am just mistaken by an intermediate. So in the right part, I am showing you the proportion of intermediates that present a particular interaction between secondary structural elements. In this case, black will be 1, meaning that it's something present in all intermediates. But as you might remember from the previous slide, this is not something that happens often, and in none of the cases in this example. So black means essentially this interaction is not present in the native state. You can see, if you look at RaptorX, for example, that you have a very important interaction between the beta strand A and the beta strand B, which is here, for example, and vice versa. This is something that has been characterized experimentally. So if you put ubiquitin in 8 molar urea, you can still look at the first beta hairpin formed by strands A and B, and you will see that it's still there and present. So it seems that RaptorX, to an extent, is producing some sort of sensible things. It's producing an unfolded state with a formed hairpin that was also identified by D.E. Shaw. So it's not entirely meaningless. But on the other hand, if you look at all of the systems at the same time, including the one DMP fold case, which I don't think you can see too well, in all of these cases, you have interactions between the alpha helix and the beta hairpin, which has not been identified experimentally. It's not present in the simulations by D.E. Shaw and doesn't seem to have any sort of real physics into it. Okay, so I realize that I'm about to run out of time, so I'm going to very quickly show you what this looks like, because it wouldn't be a proper protein folding talk if I didn't show some trajectories. This is a representative trajectory of Tia Rosetta folding, which I think, well, it's actually quite representative of the ensemble of trajectories, and also more or less presents expected cooperative two-state behavior. So this is how it looks like. So at the beginning it's unfolded, and then very, very quickly gets folded, and it has some minor adjustments. You can see that there's one loop there that moves and sort of finds its position, but the behavior is mostly two-state. If you compare this with, for example, EBFold folding, you will see a completely different thing. So we also start with the unfolded state, but then it folds in one step, then it changes again, then it reorganizes again. And even though sort of the compaction happens in one step, that's just pure physics observation, and it doesn't seem to be providing any realistic information about the folding. So after all of this, I'm just going to quickly state my conclusions. So I'm going to go for the conclusion that protein structure predictors are not telling us anything about the folding pathway. They can't tell you the kinetics. They can't tell you the rate constant. They can't tell you the intermediates. Then what are they doing? Based on also what I've shown in the case study and in the videos, I want to say that the physics that are induced by these deep learning models are unrealistic and are not correlated with the experiment. And I think something perhaps following the comments by John Molt, even though protein structure prediction might be solved, we still have a long way to go in understanding the physics of proteins, and in particular protein folding. And I would like to raise two questions, and I'll be very happy to talk about them in the Q&A. I think refolding experiments are probably the simplest way that you can think of to study protein folding. You just take a protein, you denature it, and let it renature again. It has nothing to do with what's happening in the ribosome in the cell where the protein is actually being synthesized. So I ask, if the best models that we have, if the state of the art is unable to even provide some insight into this, how can we do anything related to protein folding? We really need to start asking questions. How do we actually produce protein structures that are actually reliable in terms of folding? And another question that I have is, when we see that even though these models are nowhere near good, they do have in some cases some small signals that relate to dynamic properties. So are there dynamic properties that deep learning is trying to pick up and managing to pick up? And is there something that we can learn from there? This is all food for thought, but I'm afraid this is where my talk ends. And so I would like to thank my advisor, Professor Charlotte Dean, who is unable to join us today, and the rest of the members of my group. So Daniel Nisley, Oliver Kirk, Mark Chanowski, and Dominic Schwartz. The rest of the Oxford Protein Informatics group, the people that have funded my research, so EPSRC, Roche, and UCB. And finally, to all of you, the audience, for bearing with my talk. And I look forward to hear your questions. Thank you very much.